Hi everyone, this is Laura Slade, and I want to welcome you to today's lesson on the formation of an action potential. So the question for today is, how do nerves send messages? Well, it turns out that not just any type of message will do. The messages that the nervous system sends, they have to be fast and they have to be reliable. So today we're going to talk about exactly what type of signal it is that nerves can send, and how the basic unit of the nervous system, which is the cell of the neuron, how it's able to send that message. So it turns out that these messages, which can also be called nerve impulses, are caused by what we call an electrochemical signal. And what is that exactly? Well, electrochemical is actually Latin, and it can be translated into charged particles. So what we're talking about in electrochemical signal is the movement of small particles, mostly atoms, that have some type of charge to them. Those charged particles are primarily going to be sodium and potassium. Those are really the main players that we're going to be focusing on, although calcium also does play a role in nerve impulses. And the other thing we're going to find out today that's kind of interesting is that even though nerve impulses move down a neuron, the charged particles that we're talking about actually move across the neural membrane. That means in and out of the axon of the neuron into the interstitial fluid. So although that message, that nerve impulse, is moving down the neuron from the cell body to the terminal end, the charged particles, the sodium and potassium, are actually moving across the membrane. So the way that these charged particles move through the neuron occurs in 4 steps, and we call these 4 steps an action potential. So let's start talking about action potentials by looking at kind of a general overview of all 4 processes before we get into any specifics. So first of all, you'll see on the left-hand side of your screen showing you a cross-section of just one little piece of the axon of a neuron. So we see the cell membrane in the middle. The inside of the neuron is at the bottom of these images, and the outside of the neuron, which just kind of opens into the interstitial space, the space between cells, that's shown at the top of the image. And we're seeing this picture 4 different times as we go through each of these stages in the action potential. The other thing that we can notice in these pictures is the presence of these transmembrane proteins. So transmembrane proteins are going to be proteins that span the cell membrane of any cell. So at the start of our action potential, our neuron is going to be at resting potential. This is kind of the default stage or state for a neuron. And during resting potential, what we observe is that the inside of the cell is more negative than the outside of the cell. Then, once we have a stimulus that causes the action potential to continue in a neuron, the next stage is going to be depolarization. And during depolarization, positive particles rush into the cell, making the inside of the neuron more positively charged. Next, we are going to have a stage called repolarization, where the cell is going to return a positive charge to the outside of the cell by moving those positively charged particles back out. Finally, we observe the 4th stage of an action potential, which is called hyperpolarization. And hyperpolarization is when the cell overshoots the resting potential and actually becomes too negative. And again, all of these 4 stages are just happening because of the movement of charged particles, in particular sodium and potassium. So now we're going to go through each of the 4 stages of an action potential in more detail and find out what's really happening. And of course, we'll start at the beginning with this idea of the resting potential. So what we want to know is, how do nerve cells maintain a negative charge inside the axon? So we're seeing the negative charge on the inside of the cell. How is that maintained? Well, first of all, we have to understand that this negative charge is actually relative. which means that the inside of the axon does not have a negative charge overall. It's just not as positive as the outside of the cell. So compared to the outside of the cell, the inside is negative, even though it actually has quite a few positively charged particles. And so that is how we create this positively charged outside and relatively negatively charged inside. Now we can actually measure the amount of difference in the charge inside and outside the cell, and we do that in a unit called millivolts. So volts is a unit that you would also use to measure electricity, because electricity is also concerned with charged particles. So millivolts are a small measurement of electricity, and if we were to stick an electrode inside this axon, we would be able to measure about a charge of negative 70 millivolts during resting potential. Now the term resting is kind of in odds to what we're seeing in this phase, because what we actually know is that during resting potential, the cell has to actively spend energy to maintain this charge difference. That means that it's using ATP. And the process that actually uses that ATP is the maintenance and the running of a transmembrane protein called the sodium-potassium pump. And we see it right here in our diagram. Now the job of the sodium-potassium pump is to constantly move sodium ions out of the axon. So these are our sodium ions right here. And by the way, we abbreviate sodium as Na, and when it's an ion, when it's charged, we abbreviate it as Na+. At the same time that the sodium-potassium pump is moving sodium outside of the cell, you guessed it, it's also moving potassium inside the cell. And so we can see the potassium accumulating on the inside of the axon here. Now every time this pump operates, it's actually going to move 3 sodium ions outside of the cell for every 2 potassium ions that it moves into the cell. So because there's a different number of charged particles being moved inside versus outside the cell, that's how we're developing this overall positive charge on the outside of the cell. There are more sodiums outside than there are potassiums inside. And the reason that this process requires energy is that sodium and potassium are both being moved against their concentration gradient. That means they're being moved from an area of low concentration to an area of high concentration. And if this idea of concentration gradient is not familiar to you, I recommend that you pause this video right now and look it up either on Wikipedia or on another video on YouTube. But because the cell is using that energy to create areas of high concentration of charged particles, the cell is actually building up potential energy. This is energy that can be used later on, and it comes from the fact that both sodium and potassium want to move in the other direction through the cell. They want to go from an area of high concentration to an area of low concentration. And later on, if we allow those charged particles to move, we're going to be able to harness energy from their movement. So that's what's going on inside a neuron during resting potential, until a stimulus is introduced to that neuron. In other words, until there is a message to be carried. And then the cells are going to go from resting potential into the next stage, which is depolarization. So we can see depolarization occurring in this second illustration here. What happens during depolarization? Well, polarization, this means differences or opposites. You can think about the 2 poles of the Earth. They're on opposite ends of the globe. And we have opposites because we have a negative charge on the inside, and a positive charge on the outside. So positive and negative, these are opposites. So in depolarization, we are going to get rid of the differences. In other words, we're going to even out the charge between the inside and the outside of the cell. And we do this by allowing positively charged particles to rush into the cell. Now this will only happen when a neuron is stimulated. And a stimulation, so if you, for example, have the sensation of touch, if you have the sensation of taste or smell, what's happening is that there is a change in the charge of the axon for the particular neuron that's involved. And this charge usually has to reach about negative 55 millivolts. So we've gone from negative 70 to negative 55. So we've actually had an increase in the voltage, right? The voltage has become less negative. Once this change in voltage inside the axon happens, well, the result is that one of these other transmembrane proteins is going to open up. And this we call a voltage-gated sodium channel. There we go. So voltage-gated because when the voltage changes, the gate opens. And what does the gate do? Well, it only lets through sodium ions. It's selective. So the sodium is allowed to rush into the cell, but as of yet, the potassium can't move. And when that sodium rushes into the cell, it's going to do it through passive transport. That means it does not require energy. And this is because the sodium wants to move into the cell along its concentration gradient. So here we have the open sodium gate after a nerve is stimulated, and we can see our sodium ions rushing into the cell, making the inside of the cell more positive. And I'll just show you another view of these voltage-gated sodium channels again. And so what we see is that during resting potential, when the inside of the axon is at negative 70 millivolts, the sodium gate is closed. No sodium can get through. But when there's a change in the voltage inside the axon, so when the nerve is stimulated, the gate opens up and sodium is allowed in. So depolarization and the influx of sodium is going to continue until the neuron reaches an internal voltage of positive 35 millivolts. So we're actually going to end depolarization with more positively charged particles inside the cell than there are outside the cell. And once we hit that positive 35 millivolts inside the cell, that is going to trigger the third stage of the action potential, which is called repolarization. So we can see repolarization occurring in the third illustration here. And what is repolarization? Well, again, polarization is the difference. So what we're doing during repolarization is we are re-establishing the different charges on the inside and the outside of the cell. So that means at the end of repolarization, what we want is to once again have a negative charge, a relatively negative charge on the inside of the axon, and a relatively positive charge on the outside of the axon. And how we do this is we actually are going to allow the potassium ions to move out of the axon. And just like we had with the sodium, the potassium is going to move through a voltage-gated potassium channel. So at positive 35 millivolts, the potassium gate opens and potassium is allowed to move. And once again, this potassium is going to be moving from an area of high potassium to an area of low potassium. That means that it's moving along its concentration gradient. And because it's moving along its concentration gradient, it can do this through passive transport, so without spending any energy, without using ATP. And again, I'll just show you another image of these voltage-gated potassium gates. So what we see is while the inside of the cell is negatively charged, the potassium gate is closed and no potassium can exit the cell. But then once we hit an internal voltage of positive 35 millivolts, the potassium gate opens and potassium is allowed to exit the axon. And by the time repolarization is done, we will have restored an internal negative charge. And we see that in the third image in our set of illustrations here. So when the neuron becomes positively charged during our second phase, depolarization here, that's the nerve impulse. That is a message being carried. In repolarization, what we're doing is we are resetting to get ready for another nerve impulse, to get ready to send another image. This is like having to set the dominoes back up after you've knocked them all down. So if we have set up the dominoes, if we've returned an internal negative at the end of repolarization, well then what's left for phase 4? Well, the fourth and final stage of an action potential is called hyperpolarization. And during hyperpolarization, what we observe is that the cell overshoots resting potential. It actually ends up becoming too negative, more negative than it wanted to be inside the axon. And the reason that we observe this is because it turns out the potassium gates kind of end up closing slowly. So they would like to snap shut at negative 70 millivolts perhaps, but because they're not too quick on their feet, takes them a little longer, a few extra potassium are allowed to rush out, and that causes the inside of the neuron to go all the way down to negative 90 millivolts, which is more negative than our resting potential. And I'm going to explain to you in a second why this might not actually be a bad thing. But first, there's something I want to point out, and that's the fact that at the end of an action potential, we have indeed returned the negative charge inside the axon. So we saw that here in repolarization. However, what has happened is that the sodium and potassium ions have actually switched locations. We started with sodium on the outside, potassium on the inside, but we've ended with potassium on the outside and sodium on the inside. And so even at the end of hyperpolarization, we actually still have to return the axon to resting potential. And that is done again by the sodium-potassium pump. So over time, and we are talking milliseconds because nerve impulses are sent at an incredible rate in our bodies, but in terms of the length of a whole action potential, it takes time. The sodium-potassium pump is going to restore the sodium to the outside of the cell and the potassium to the inside. But once that's done, once the sodium-potassium pump has been operating for a little while in order to switch those ions back around, then we're back at the beginning. We're back at resting potential, and this nerve will be ready to send another impulse or send another message. Now, we said at the beginning that you could actually stick an electrode into a neuron and measure its voltage in millivolts. And actually scientists have done this with the axons, so the nerves, from giant squids. And when you do this, stick an electrode inside an axon and measure its voltage, you would get a graph like the following. So on the x-axis, we're looking at time. So this is an action potential that's happening over time. And again, very quickly, happening in milliseconds. And over time, what we're doing is we're reading that membrane potential, so the charge difference between the inside and the outside of the cell, and we're measuring that in millivolts. And what will we see if we did this? Well, we would see that at the beginning of an action potential, so during resting potential, the charge inside the axon would be about negative 70 millivolts. Then the neuron needs to be stimulated. So we need to somehow cause the inside of that axon to start becoming a little bit positive. And if it becomes positive enough, it is going to cause depolarization. Now, this idea of becoming positive enough, we call that the threshold level. And I'm going to talk to you more about the threshold level the next time that we get together. But assuming that that stimulation happens, then what we're going to see next is the neuron go into depolarization. So as you can see here, the voltage is going to increase while the neuron is being depolarized, and that's because those sodium ions are rushing into the axon. Then, at about positive 35 millivolts, the sodium gates are going to close, and the potassium gates are going to open. Once the potassium gate opens, the third stage of an action potential is going to occur. That is repolarization. So the inside of the axon will be becoming negative again as the potassium rushes outside of the axon. In the final stage of the action potential, we're going to see that too much potassium leaves the axon, and that is going to cause the axon to become more negative than it was to begin with, and we call that hyperpolarization. So we can see here that this hyperpolarization is going to bring the charge of the inside of the axon all the way down to about negative 90 millivolts. Then that sodium-potassium pump is going to start working again, and it's going to return the axon to its resting potential so that it's ready to send another impulse. And we see that here, back to resting potential and ready to send another message. Now, why might hyperpolarization actually be a good thing? Well, it turns out that during hyperpolarization, when the axon here is too negative, it's actually not very easy, in fact, it's basically impossible to stimulate the nerve again until it's got back up to resting potential over here. And this period of time when the neuron cannot be stimulated is called the refractory period. And there's a few good things about having this refractory period. First of all, it helps our nervous systems from becoming overwhelmed. So can you imagine if your nerves were firing all the time at every millisecond? It's too much information for the nervous system to process. Also, we're going to find out that this refractory period, where that section of the axon cannot be stimulated, also helps with the one-way travel of these nerve impulses. And that is something that we'll talk about the next time we get together. So we've talked about what type of signal nerves actually send. We found out that's an electrochemical signal. We figured out how neurons are actually capable of sending those messages, and it turns out they do it by moving charged particles across their cell membranes. And those particles are sodium and potassium that they're moving in and out of the axon in order to change the charge inside the axon and send a message. So leave it there for today, and I'll see you guys next time.